Hi everyone, this short video is going to cover the last 3 steps of glycolysis to finish off our chapter on glycolysis. This is step 8, where we see a conversion of 3-phosphoglycerate to 2-phosphoglycerate. And what's happening here, if we look at the configuration on the left for 3-phosphoglycerate, we see that the phosphate is on the 3rd carbon. So we count from the top, carbon 1, carbon 2, carbon 3, and the phosphate group, which is a P with a circle around it, is on the 3rd carbon. And we're actually having an interconversion of where the phosphate group is. If you look at the picture or the structure on the right, we see that the phosphate is now on the 2nd carbon. So this is the inner transfer of a phosphate group. So the substrate is 3-phosphoglycerate. The product is 2-phosphoglycerate. They are isomers of each other, so it's merely an isomerization reaction. The enzyme required here is a mutase. So most of the time we would think it would be an isomerase, but it's actually a mutase because we're having an internal transfer of a phosphate group. So a mutase does the internal transfer of a phosphate. So the possible enzyme name is 3-phosphoglycerate mutase. So the actual enzyme name is exactly what we predicted, which is phosphoglycerate mutase. Again, the delta G, or the Gibbs free energy for this reaction, is a small positive at 4.4. Therefore, this reaction is spontaneous. As well, we see bidirectional arrows, so it is a reversible reaction. So this is a spontaneous reversible reaction. Step 9, our product from step 8 becomes our substrate for step 9. So our substrate is 2-phosphoglycerate, and now the product is phosphoenol pyruvate. And here we see that water is being moved out of the reaction. So we're going from one substrate to one product with the removal of water. That would be a dehydration reaction. So there's 2 biochemical processes occurring here. The first one is a loss of water, and the second one is not as intuitive, but it's a breaking of a carbon-oxygen bond. And so you can see this. I've indicated it with a red arrow. So on the left, we see that there's a hydroxyl group on the 3rd carbon. And on the right molecule, we see that that hydroxyl group is gone. So we see that the bond between the carbon and the oxygen is broke to release the water, and therefore it's a breaking between the carbon-oxygen bond. The enzyme that's required for this is a lyase, because we're naming it based on the second biochemical process, which is the breaking between the carbon-oxygen bond. So the possible enzyme name is 2-phosphoglycerate lyase. So for this reaction, the possible enzyme name is not the actual enzyme name. The actual enzyme name is enolase. And then another name for enolase is 2-phosphoglycerate hydrolyase. And this encompasses both the dehydration reaction and the breaking of the carbon-oxygen bond. As we can see, the Gibbs free energy is a very small positive number, and so this reaction is spontaneous. And as a result of the bidirectional arrows, it also is reversible. Now we're finally at the last step of glycolysis, where we see phosphoenol pyruvate getting converted into our final product of glycolysis, which is pyruvate. Remember, this happens twice, so we do have 2 pyruvates produced from one glucose molecule. So the substrate here is phosphoenol pyruvate, which we see the chemical formula, or the chemical structure rather, on the left at the top. And then the product is pyruvate. What we see going into the reaction is ADP, adenosine diphosphate, and we see ATP, adenosine triphosphate, being produced. So something is losing a phosphate. And so if we look at the chemical structure on the left-hand side versus the chemical structure of pyruvate on the right-hand side, we can see that pyruvate is missing a phosphate. So the biochemical process here is dephosphorylation The possible enzyme could be a phosphatase, but we know that we name it in the direction that ATP is being added. So it's always going to be a kinase. So the enzyme required would be a kinase, not a phosphatase. So the possible enzyme name, based on our naming rules, would be pyruvate kinase, because we always name in the direction that ATP is being added. So in this case, it would be in the reverse direction of the formula. So we are correct in our naming. We always name in the direction that ATP is being added to the reaction. So in this case, it would be the opposite direction. And so the enzyme is pyruvate kinase. This is a highly exergonic reaction, so we actually release a huge amount of energy. Therefore, it is a large negative and spontaneous. As well, we can see there's only a unidirectional arrow, and therefore it's an irreversible reaction. Remember, the first reaction of glycolysis is irreversible, and the last reaction of glycolysis is irreversible to prevent the metabolic pathway from being able to be reversible and nothing getting done. Okay, so this is the last step of glycolysis. The reaction is irreversible to prevent the metabolic pathway from being reversible. So here is a summary of glycolysis. Remember, go back through your notes and summarize the inputs and outputs of glycolysis. You can also look at the chemical formula, which is on the top of the slide in red, where we have glucose plus 2 ATP plus 2 coenzymes, which are NAD, creates 2 pyruvate plus 4 ATP plus 2 NADH. So the inputs, therefore, are 1 glucose, 2 ATP, 4 ADP, 2 NAD+. And then the outputs are the products, as you can see here, are 2 pyruvate, 4 ATP, 2 ADP, 2 NADH, which we'll see goes to the electron transport chain, and 2 water molecules. So to summarize glycolysis, first of all, we need to remember where it happens. And it happens in the cell cytoplasm, or the cytosol, all the time. Hundreds of thousands of times every second, to ensure that we have enough ATP to sustain all the mechanisms that are happening in the cell. Why does it happen? Well, it is the first step of glucose oxidation. So we're creating ATP from glucose. What is involved? Well, it's a process that splits glucose into 2 pyruvate, 2 ATP, and 2 NADH. The NADH then travel to the mitochondria, and as we'll see in a few weeks, it enters the electron transport chain and yields a huge amount of energy. So remember, glucose oxidation happens with blood glucose or through glycogen, which is our stored form of carbohydrate within the cell. So it really depends whether we're fed or fasted, whether we're at rest or exercise, will determine the source of the glucose, whether it's coming from glycogen or coming from our blood. So when does this happen? Well, as I mentioned, it happens all the time. And the rate of glycolysis really depends on the demand for ATP within the cell. So remember, we talked about the supply-demand relationship. And the energy status of the cell will dictate how quickly we go through glycolysis to meet the demands of the cell. There are 9 to 10 enzymatic reactions of glycolysis, so it's a relatively fast metabolic pathway compared to other processes within the cell. And really, the energy status of the cell, the AMP-ATP ratio, will determine the flux or the rate through the pathway.